All right, I'd like to talk to you a little bit today about bacterial genetic variation. So bacteria are probably the most diverse set of species that we have on this planet, right? With a clade, so for instance, if we took E. coli, there's many different species of E. coli because they have so much genetic variation. So all the different species of that single E. coli would be called a clade, right? So things that we know about bacteria, right? They have a singular chromosome containing all the genes, right? All these housekeeping genes are in the one circular chromosome they have. But we've said that they also have these small circular pieces called plasmids, right? And the plasmids include genes for survival. Right? So genes necessary for survival. Right? And we're going to talk about these genes for survival can be passed between different E. coli, right? There's a couple different ways we'll talk about. We'll talk about transformation, conjugation, and transduction. So, well, we'll go through all three of those methods. All right, so transformation. So this was the first one discovered, right? Very famous experiment. A guy named Griffith, right, in 1920, was he was looking at strep pneumonia, right? So if you have a strep pneumonia, that's what Griffith was looking at, right? And so he did this experiment. So he took a healthy mouse. So if you look at the picture, right, the first one on the left, he took a healthy mouse and gave it rough, non-virulent strain, right? Then he injected it in the mouse, and the mouse didn't die. Nothing happened. Then he took a smooth strain of the same bacteria, right, and injected it into the mouse, and the mouse dies. So this is called the virulent, right, or the disease-causing strain, right? So then he took the virulent strain and he boiled it, right, and then after it cooled off, he injected the mouse, and nothing happened, right? So heat kills bacteria, and we all know that. That's why we heat sterilize things. Right, so then he did one other part of the experiment. He took and mixed the non-virulent or the rough textured bacteria with the heat-killed smooth bacteria. Then he mixed them together, right, and then he injected them into the mouse. And then the mouse dies, but then the resulting bacteria that are left, when he looks at the mouse, they're all smooth, right? So he said that something must have happened to transform the rough bacteria into smooth bacteria, right? And we're going to talk about transformation. We use transformation a lot, right? So make sure you know the experiment, but also that transformation is the process where bacteria pick up DNA or plasmids, right, from the environment. Right, so usually this happens in time of stress, right? So when this mouse's immune system is trying to fight off the rough, non-virulent bacteria, they're looking for pieces of DNA they can get. Right, so conjugation, right? So conjugation is another one, right? So certain bacteria, not all bacteria can do conjugation, but certain bacteria can. So E. coli is one, and E. coli we study a lot, are able to transfer bacteria, right? So this, if you look at the picture down here, I'll make it just a little bit larger. Right? Then I'll try to move it over a little bit. All right, so if we look at this picture, right? So the one on the left, so the E. coli on the left, right? Remember E. coli is a bacillus, so this is E. coli. Right? So E. coli can form what's called a sex pilus, right, which is essentially a cytoplasmic bridge. And when it forms that cytoplasmic bridge, right, it can transfer plasmids from one organism to another. So if you look at this one on the left, see all the little spikes coming out? Those are all attempts to find another E. coli. So if they don't have eyes, they can't see this one. But once they come in contact with another E. coli, then they'll connect that bridge, right? And they'll start passing plasmids. And this is usually in times of stress, right? E. coli has no brain. It's not thinking, well, this is for the good of the species. But when you stress it, it tends to find other things to pass its plasmid to. Remember, plasmids can have things like antibiotic resistance genes. It can have genes for digesting other food. But ultimately, the survival of the species of E. coli is better if there are more E. coli, because your immune system is less likely to kill all of them, right? Right. So a couple things, right? So F, right here, is called fertility factor, right? So in order to participate, right, we say that the E. coli must be F positive. So if you look at the picture on the right of that, the E. coli on the left is F positive and has a plasmid. It can form that sex pilus or the cytoplasmic bridge. And once that's complete, right, it can transfer a copy of its plasmid, right, to the other one. Now the other one now will be F positive, right? And that new E. coli that's now F positive can find and transfer it to other. Once it makes a copy, they can transfer it to other, right? So that's why we say that all of our bacteria that we start out with in lab are BSL-1s, but they can become BSL others, right? Because we all have bacteria, and chances are that we've all used antibiotics at some point. So we all have bacteria that might be antibiotic resistant, right? Transduction is our third method, right? So now we're 1951, right? Was discovered when researching salmonella. So now salmonella doesn't have fertility factor. So salmonella can't transfer via conjugation. Transformation will work for it. Transformation will work for essentially any bacteria, but conjugation will not. But they studied it, and they were trying to figure out how these plasmids and these DNA got transferred between salmonellas. And they found that it was through a phage vector, right? Remember, viruses are bacteriophages, right? So DNA that's transferred through a phage is called transduction, right? So interesting thing, and I'll introduce it now. So with eukaryotes, transfection, right? So transduction for bacteria, but if you have a virus, right, and you replicate that virus, and then that virus comes and infects me, that's transfection. Or if I genetically engineer a virus and purposefully infect cells with DNA, right, that's transfection, right? So transduction for bacteria, transfection. So this bacteria on the left is producing virus, right, and they're producing that protein coat, that tail, that head that contains the nucleic acid. When it releases the phage, it finds a new one and injects its nucleotide in, and it becomes part of the host DNA, right? All right, and then restriction enzymes. We've already talked about restriction enzymes, right? Discovered in the late 1960s. So we've gone from 1920 to 1960, 40 years, right, of research. So early 1970s, a graduate student at Stanford, right, smart school, proposed that you could use restriction enzymes to engineer plasmids, right? So in 73, Herbert Boyer, right, published a paper, right? So the graduate students working for Herbert Boyer. In 1976, Boyer founds Genentech and begins making insulin from E. coli. Right, less than, well, just about 14 years later, he sells Genentech for $46 billion, right? And now Biotech's on. Right, these small discoveries, right? These small discoveries changed the world, right? And they made for great financial success. All right. So things that you need to know about plasmids, right? Things that plasmids should have. So they should have an origin of replication, right? Because ultimately, we want bacteria to be able to replicate this plasmid and pass it to other bacteria, right? When bacteria reproduce via binary fission, right? The new bacterium should have this plasmid in it. All right. So origin of replication. They should have a promoter region. So remember a promoter, right? Let me make this just a little smaller. So a promoter, right? So it's the site of RNA polymerase attachment, right? So this is what's going to make the mRNA. Right, so that's the site of the RNA polymerase attachment. So it needs a promoter site, right? It needs a insertion site. So this is an insertion site. Right, it's insertion or a cloning site. Right, so just to piggyback off of that, remember an MCS, which is a multiple cloning site. Has many restriction sites. So it has a whole host of palindromes, right? So I want my cloning site. I want a terminator sequence of some sort, right? So something that tells it to stop transcription, right? And that'll be kind of at that 3' site that you see on that right there. I want a selective marker, right? So a selective marker is here. So the selective marker allows for identification, right? Allows for identification of transformed bacteria. So those bacteria glow green, or I can put a red marker, they could glow red. They can make the colonies blue. So it really is some sort of color, right? Right? It could be RFP, right? And then an antibiotic resistance. We always give it an antibiotic resistance gene as well, right? So there's my selectable marker, my antibiotic resistance. So ampicillin is the most common. So there's two varieties. There's the AMP-R gene, so it's ampicillin resistance, and there's the BLA gene, beta-lactamase, and that what that does is it deactivates ampicillin. So the BLA or the AMP, they're both acceptable, but they all have to have those components. All right, so short ed puzzle, but it gives you all you need to know for today.